So hello friends, welcome to another video on plant virology. So as you have already know that in this series we are discussing about the unit 6 as per the ASRB syllabus for the NET, ARS, SMS or STO. The unit 6 plant virology we are discussing. So we have already covered the nature, composition and architecture of plant viruses. These things we have already covered. Now it's time for the viroids. So today we will discuss about everything about the virus, starting from the nature, composition, architecture. In addition to this, we will also discuss about its localization in the plant cell, then about its movement, its transmission, then about the classification of plant viroids, then about the further about the detection methods used for the plant viral detection, and ultimately the management practices for this pathogen. Okay, because here in this total syllabus for the plant virology, you can see here that no other single sentence mentioned about the viroid apart from this. So whatever we will cover, it's better to cover all this thing about viroid in this single video, as we might forgot in the later the later time. Okay, so we will cover everything about viroid in this single video. So without further delay, let's start the video. So we have discussed about this general characteristic of virus, then composition of virus we have discussed, architecture of virus we have discussed. If you want to know, you want to study in detail about all these things, you can check the previous videos. Okay, so let's start about the viroid. So first about the definition. So viroid are the smallest known infectious agent of plant. It is the smallest known. You can call it as the smallest pathogenic agent that cause disease in the plant is the viroid. Next, it is subviral agent. Then it is acellular. Just like virus, it is acellular. Then it consists of naked single-stranded circular RNA. Remember this. So it is containing a naked single-stranded circular RNA, covalently closed circular single-stranded RNA without any protein code. So main difference between the virus and the viroid is it lack protein code, or it is not having any capacity to code the protein. It lack protein coding capacity. Okay. Another thing I forgot here to mention, that the first viroid or the first plant viroid discovered is potato spindle tuber viroid. For viroid generally VD was written. Okay, you know that for tobacco mosaic virus short abbreviation form is TMV. V is there for virus. Similarly, for mentioning the viroid, VD is used as abbreviation. So the first viroid reported was potato spindle tuber viroid. Who discovered this? T.O. Diner. Okay, and remember the year also, that is 1971. Okay, you have to remember this. This is very important and most frequently asked question for various exams, not only the ARS or NET exam. In most of the exams, entrance exam, it was asked. So the first viroid discovered was potato spindle tuber viroid by T.O. Diner in the year 1971. So definition we have covered. Next, next, this is the structure of the viroid. This is a naked single-stranded circular covalently closed RNA molecule. The general characteristics of the viroid. So if it is asked in 10 marks or 15 marks, you can mention this, or you can shorten this for the 5 marks also. So it's a subviral agent and subviral acellular agent that infect plant only. No virus has been reported to infect the humans. Next, it is the smallest known infectious pathogen of plant. Remember this. This was multiple time asked in the entrance. Okay, this is important. Next, genome. Genome is naked, single-stranded, circular RNA. Okay, never, no DNA is there. Never, DNA is not there for the viroid. It is single-stranded circular RNA. Then genome size is 246 to 401 nucleotide. So this 246 is for the coconut kadang viroid, the smallest known plant viroid whose genome size is 246 nucleotide is coconut kadang viroid, and the largest viroid whose size is 401 nucleotide is Chrysanthemum chlorotic mottle viroid. Okay, Chrysanthemum chlorotic mottle viroid. So these are two, the smallest and the largest viroid known for the plant. Okay, remember this. This is actually very important for the MCQ based question. Then I have already told you it lacks protein coat, envelope or capsid. Then it also lacks protein coding capacity because it lacks ribosome and enzymes. Then it is this is the very important point. It is high, it's so high cell complementary RNA forming rod like secondary structure. See, it is not having any protective coat outside it, means it needs something, some mechanism it needs to protect its RNA, because if you are working in a molecular lab you might know that RNA is very fragile thing, it degrades very easily. Then how a viroid survive in the nature? Because of this complex secondary structure. Because of formation of the secondary structure, it is actually showing resistance to nuclease attack, because RNA will be degraded by the nucleases. So by the formation of secondary structure, it resists the nuclease attack and becomes stable. This is very important. Okay, in the assessment reasoning type question, this might be asked. So remember this. Okay. Next, the point number 8. It's a obligate intracellular parasite. Means it will replicate inside the host cell, not in the intracellular space or not outside the host cell. It will, just like the virus, it will replicate inside the host cell using the host enzyme because it lacks the enzyme activity. Next, the ribozyme activity present in some viroid. In some viroid like the like our Apsun viroid, we will discuss in the later slides. Okay, so avocado sunblotch viroid, so there they have ribozyme activity because they have branching, so they need to be cut. For cell cleavage, this ribozyme activity is present for some viroid. Then the most important thing, movement of plant viroid. This we will discuss in another side in detail. Okay, just for your knowledge, I will say that this small viroid actually pass from one cell to another cell through the plasmodesmata. Okay, for the spread of disease. So this is all about the general characteristic of the virus. So let's revise. So it's a subviral agent. Okay, smallest known infectious agent. Genome is single stranded circular covalently closed RNA. Remember, RNA. Then genome size ranges from 246 to 401 nucleotides. Then it is lack protein coat, no protein coding capacity. Then it shows high self-complementary forming rod-like secondary structures. It's an obligate intracellular parasite. Some viroids are having ribozyme activity and it moves from one cell to another cell through plasmodesmata. Okay. Next example of viroids, important viroids. Okay. So potato spindle tuber viroid. This was the first viroid reported in plant by T.O. Diner in 1971. Okay. Next, citrus exocortis viroid, Hobson viroid, chrysanthemum stunned viroid. These are few viroids. Okay. So this is a quick summary. The viroid are circular RNA plant pathogen without any protein coat or coding capacity, replicate using host enzyme, moves through plasmodesmata, cause distinct plant diseases. Okay, this is brief about plant viroids. Next, about the structure of viroid. As you all know, this is a naked, circular, covalently closed, single stranded RNA molecule, and it is forming rod-like structure, secondary structure by cell complementary. Because it's so some cell complementary. Cell complementary means the sequence present inside the RNA, they will join. Okay, they have the complementary sequence, so they will join, forming this rod-like structures. Okay, this is just say like that. So this is not a perfect rod, this is quasi rod shaped. Then so the viroid is having 5 domains. So what are the 5 domains? First domain is the TL, means terminal, left terminal domain. So left terminal domain, why it is important? It is important for the replication and host interaction. Then second domain is the pathogenetic domains or P domain. So what's its role? Its role is for symptom expression. Then third and most important domain is central conserved region, CCR domain. Its role is replication and processing of the RNA molecule. And it is characteristics of the post-viroidy. CCR region is absent in absent viroidy. Okay. So for your knowledge, I want to tell you that 2 family of viroids are there, post-viroidy and absent viroidy. We will discuss in the later slides. So this CCR region, central conserved region, is the characteristics of post-viroidy. Next important region or domain is V domain or variable domain. This is for the variability, sequence variability. Next, the final 5th domain, right terminal domain. It is also for the host specificity and movement. So this is about the 5 domains present in the viroid RNA or viroid structure. Remember this. Okay, this is important. Then structure feature we have already discussed that it is uncapsulated, no protein coat, single stranded RNA molecule, so cell complementarity, forming rod-like structure, and it is having functional domain. These 5 functional domain it is having. So if it is asked in the ARS mains exam, you can draw it for 15 or 10 marks question, and you can simply mention these things. This will look very good, and this function of this domain you should remember, so that it will be helpful for your MCQ based question. Okay, remember these things. Okay, in short it is written. Next, about the composition of viroid. So we have already discussed that it is only the RNA, single stranded circular RNA, with and the size ranges from 246 to 401. High self complementarity is there, high GC content. Remember this, around 55 to 65%. The GC content is around 55 to 65%, and the size of the viroid is generally 37 nm. These are important thing. Okay, remember this numbers. The percentage of GC content in viroid, then size of the viroid 37 nm mostly. Okay, so it is lacking protein, no enzyme, no lipid, no water, no iron. So you can compare this with virus. In virus, all these things are present. In viroid, only you have to mention these things. If for in mains exam, if it is asked that write the composition of virus or viral, you can simply remember these points, so you can write it. Okay, next, about the architecture of viroid. So the virus architecture was very actually complex. 3 types of symmetry were observed, but here this is simple circular single stranded RNA. So already we have discussed naked RNA highly folded, forming quasi-rod shape, and the stabilization by intermolecular base pairing. Okay, due to the self-complementarity. Okay, so no such architecture is there, just like the virus. Okay. Next, most important thing, localization of viroids in plant. So first thing is how you will localize a viroid. So the method to localize the viroid is by using in situ hybridization method. Method R, in situ hybridization method, confocal laser scanning microscopy method, or using the TEM or transmission electron microscopy. So these are the method to study the location or localization of viroids in plant. So in general, viroids located inside the nucleus. I have already told you that viroid is a obligate intracellular parasite, means it will localize inside a cell. So inside the cell, where it will localize? It will mostly localize in the nucleus. And if you see the tissue, then mostly it is found in vascular tissue and sometimes also found in mesophyll tissue. Okay, this is about the tissue distribution and general location. Then inside the nucleus, nucleolus is there and nucleoplasm is there. So the viroid, like potato spindle tuber viroid and coconut cadang virus, these two viruses mainly localize in nucleolus, but the citrus exocortis virus, this actually predominantly accumulates in the nucleoplasm. Remember this for the MCQ types question. Okay, this is very important. Which viroid localize in nucleolus? For nucleus, it is potato spindle tuber viroid and coconut cadang virus. Both are very important. And the citrus exocortis, this actually accumulate in nucleoplasm. And major exception means the viroid that not localize in the nucleus. Where it will localize? It is localized in the chloroplast. So the viroid avocado sunblotch viroid, it is the exceptional case that is localized in chloroplast. So as it is localized in the chloroplast, means it is associated with some chloroplast abnormalities. Okay, for your memory, this is summary. Most viroid generally located in nucleus. Potato spindle tuber and coconut cadang located in nucleolus. Citrus exocortis in nucleoplasm, and avocado sunblotch viroid, which is an exception, located in chloroplast. Okay. Next, about the movement and transmission of virus. So virus movement means how it spread from one cell to another cell, but it is inside a single plant, that we will call movement. But transmission means how it transmits from one plant to another plant, one location to another location totally. Okay, not one cell to another cell. So first the movement. So movement of plant viroids generally occurs through the plasmodesmata, I have already told. Then how it mediate the movement? So movement is mediated through a specific RNA sequence or structural motifs. I have already discussed that some regions are also there or domains are there that is helpful for the movement. That is our terminal right domain. This is for the movement. Okay, this region of the viroid also helpful for the movement of plant viroid. Apart from this, some host protein also helpful for its movement. But like the viruses, it is not containing any type of movement protein. In the virus, we will later discuss that how plant virus moves. So many strategies are there. For potyviruses, different strategies there. For Gemini virus strategy is there. So helper strategy, then nuclear shuttle protein strategy, movement protein based, tubule protein based. So many strategies are there for the movement of plant virus from one cell to another cell. So we will discuss later. But for the viroid, there is no such mechanism is there. It will just mediate by some RNA sequence or some using some structural motifs. Then this is for the short movement, means one cell to another. If it has to travel long distance inside a plant, then it will systemically move through the phloem tissue for long distance movement. Okay. Next, the factor facilitating the movements are, first most important thing is viroid shows high resistance to nuclease. Why this viroid RNA is showing high resistance to nuclease degradation? I have already told, because of the presence of complex secondary structure. Because of the presence of secondary structure, it is showing the resistance to nuclease degradation. Next factor facilitating the movement is the virus translocation is also favored by some host proteins. Okay. Next, about the transmission of viroid. So generally viroids are transmitted by mechanical means. No vector is involved like the viruses. No insect vector, no fungal vector, no nematode, nothing. Just by mechanical transmission, it transmits from one host to another host. So by contaminated tool or knife or pruning equipment or by grafting, it transmits from one host to another host. But some cases vertical transmission is also observed. Like some viruses are pollen and seed transmitted, like observed in tomato, potato and grapevines. So this transmission is affected by both the structure of virus as well as host component. Because of the presence of secondary structure, it resists the nuclease degradation, and in complexing with some host proteins, it actually enhances stability and further helpful for the transmission. So in summary, for the movement, cell to cell movement occurs through plasmodesmata, and for the long distance movement, it is mediated by the phloem. Next, the viroid is mostly transmitted mechanically, and few viroids are also transmitted through pollen and seed. Next, about the classification. This is very important actually. So viroid are classified into two major families, Postviridae and Absonviridae. So this is the major family, and this family is very small, contain only few viroids. So the most important one are potato spindle tuber. The size also you have to remember, 356 to 395. The citrus exocortis, hopstern, then coconut cadon 246 is the smallest viroid. Okay. Then apple scat skin. So for few virus, you should remember the size also. It might be asked in the exam. And avocado sun blotch, so size is 247. Then chrysanthemum chlorotic mottle, size is 399 to 401. I have already told this is the largest viroid and this one is the smallest viroid. Okay. For few viroid, you should remember the size, That will be helpful for MCQ based question. Okay. Next, how this family are formed. So major differences are there. So we'll discuss that. So first, the type species. For Apson viroides, the type species is avocado sunblotch viroid, and for possibility it is potato spindle tuber viroid. Then the major characteristic feature, the central conserved region. I have already told the viroid is containing 5 domains: T domain, then P domain, P for pathogenicity, T for terminal right, then this variable domain, then central conserved domain, and terminal right domain. So this central conserved region is absent for the possibility, whereas it is present or characteristic for the possibility. Then hammerhead structure. Hammerhead is nothing but the secondary structures. Highly folded secondary structure is present in absent virility, which is absent in possibility. Because of presence of second hammerhead structure, it needs ribosome activity during the replication, because it needs to cut that portion, to cleave that. For cell cleavage, it needs the ribozyme activity during replication. So this is present, ribozyme activity is present in Apson viroides. But as the CCR, as the hammerhead structure is absent for the possibility, it is not requiring any type of ribozyme activity, so it is obviously absent in the possibility. Then site of replication. We have already discussed in the localization part that the Apson viroid or avocado sunblotch viroid localize in the chloroplast. This is the exceptional viroid. So the site of, because localized means it is replicated inside that only. That's why they are localizing at that particular region. So the site of replication for the Apson viroid is chloroplast, whereas for the possibility viroid it is nucleus. The replication mechanism, symmetric rolling circle, whereas for the possibility it is asymmetric. So what happened? Due to the presence of hammerhead structure in the Apson viroid, it needs two rolling cycle for the replication to complete. That's why it is symmetric. Whereas in possibility, no hammerhead is there, no ribozyme activity or cell cleavage activity is needed. So only in one rolling cycle the replication is completed. That's why it is asymmetry. Okay, here 2, here 1. Actually, for just like the virus replication, you know that this circular kind of nucleic acid generally replicate through the mechanism of rolling circle model. So here it is for the viroid, this nucleic acid is circular RNA. That's why it also follow the mechanism of rolling circle for its replication. Then the secondary structures, it is highly branched, but this is rod-like linear type. Then host end, this is narrow. This is for the possibility is broad. Then symptom, usually for Apson viroid it is mild to or symptomless, but wide range of symptoms are produced by possibility. These are the examples. Okay, this is very important table. You should remember this. This is very, very important. Multiple type question, as well as for the mains exam, for every exam. This is very, very important. Every example you should remember. And this difference, but the like the presence of CCR region, ribozyme activity, then the site of replication. These are very important. Okay, you can read this memory aid, like Apson viroid means chloroplast plus hammerhead plus ribozyme plus no CCR, whereas possibility means nucleus plus CCR, no hammerhead, no ribozyme. Next, the diagnostic procedure for viroids. So it is very difficult to detect the viroid. Why? It is not having any characteristic particle, or it is not having any protein. So no immunological method like ELISA or serological method can be used, or any because it is not having any crystal structure on any characteristic particles. So electron microscopy also can't be used for the routine diagnosis. So here only a circular naked single stranded RNA is there. So whatever nucleic acid based detection method are there, that can be used. So the major diagnostic methods for the viroid detection are: first, the biological test using the indicator plant. Indicator plant means those plants which produce some specific symptoms when that particular pathogen will be infected. Okay, by using the symptom expression studies, we can use some indicator plants for biological tests. Then the advanced tests are gel electrophoresis. So we can do the gel electrophoresis like normal agar gel electrophoresis can be used. And most important, R page, return page. What is return page? We will discuss in later videos. This will be otherwise very long video for us. So we will discuss it later. So R page is mostly used for the viroid. Then the PCR or RT-PCR can be used, and nucleic acid based hybridization method. So in the laboratory technique portion, we will discuss all these methods in very detail. Okay. So these are the methods for the detection of viroid. Next, final slide, the management of plant viroid diseases. So like the viruses, here for the viroid also no management strategy is there. Here only you can do the prevention or exclusions. Okay, so you can use the viroid free planting material, roguing can be done, then transmission management like means proper sanitation can be maintained, then use of meristematic tip so that you will get the viroid free culturing material. Then about the sanitation hygiene of the equipments or tools, then the quarantine measures to restrict the movement of infected planting material, then resistant or mostly the tolerant cultivar can be used. No such resistance are there, so tolerant cultivar can be used, and using certification program for regular indexing and molecular testing of the planting material or mother stock maintenance. So these are some preventive measures can be done to avoid the plant viroid disease. So this is all about viroid. I think you people are very much clear about viroid. We have discussed everything about viroid. Okay. So if you have any doubt, you can mention it in the comment section. I will try to clarify your doubts in the further videos. Okay. So I hope this will be helpful for you people for ARS NET or for other exams. So thank you for watching this video.